HASEENA RAFATH, Assistant Professor, Department of Botany, Annamalai University. Today we are going to discuss a topic called physical methods of gene transfer. This topic comes under biotechnology and genetic engineering for second M.Sc. Botany. So, we use these methods, these are the direct gene transfer methods. These methods we are using to escape the host range limitations, and the gene can be transferred directly without using any biological vectors into the plant cell. And because we are using these methods, we have cell wall as an efficient barrier. So, we are using the protoplast as a particular plant cell in which we have to transfer the gene. So, we are using the naked plant cell or protoplast, found best experimental system for the study of these methods. So, this is also referred to as DMGT, that is a DNA mediated gene transfer method. These methods we have nearly from 9 to 10 methods, but mostly we are using 4 to 5 methods in all the biotechnology industries. Only we are using these 4 commonly used methods. First one is electroporation, second one is microprojectile or biolistics or particle bombardment. The third one is liposome mediated gene transfer, and the fourth one is microinjection. So, the first one, that is the electroporation. This method was first invented by a scientist called Newman et al. And his group in 1982. This method uses the electrical impulses. These electrical impulses of high field strength are used, and so that the permeable cell membrane can uptake large molecules including DNA. The size of the DNA can be 1 to 1.5 kilobases. Initial field strength is used with the low capacitance and therefore a short decay time. Now, we discuss about the process, how this electroporation can be done with the help of the protoplast. In this procedure, a sample of protoplast is used and it is passed through a high electrical pulse in a chamber of electroporation. The chamber is cylindrical in form with a distance of 1 centimeter parallel steel electrodes. The pulses is applied by the discharge of capacitor across the cell. And here we are using polyethylene glycol, so that the transformation frequency can be increased. This PEG can increase the insertion percentage of the DNA into the protoplast. The DNA, that is the foreign DNA which we are isolating from another plant, and we have to insert this into the plant cell of the another plant. So, PEG is believed to assist the association of DNA with the membrane. So, this is the process, and we have certain advantages and disadvantages of this method. First we will discuss about the advantages. This method is convenient, very simple and fast, and has low cell toxicity. The toxicity of the cell will be very low, and easily the DNA can be inserted into the plant cell or the protoplast. How we are able to insert the foreign DNA into the protoplast? Because we are passing high altitude or high field strength electric pulses. So, these electric pulses or electric waves will create pores on the protoplast, so that from the pores the DNA will get inserted into the protoplast and the formation of our DNA takes place. And the disadvantage of the technique is difficulty in generating plants from protoplast. See, because we are using this direct method, since we are using the electroporation method, because we are passing the high electric impulses into the PEG liquid of the electroporation, what happens is most of the protoplasts are getting damaged. Only few protoplasts in which the DNA is getting inserted, and the percentage of viable cells are very low. So, this is the main advantage why we are not able to use it very frequently. Next one is liposome mediated gene transfer. In this we are using liposomes from the polioviruses of the RNA. So, first this liposome method was used for the delivery of the DNA in the poliovirus. The use of liposomes of transformation is called as lipofection. When we are using this method in the viruses, so we call it as lipofection. Liposomes are artificial lipid vesicles surrounded by a synthetic membrane of phospholipids. This membrane have been used in mammalian tissue culture to deliver drugs, proteins, etc. into the cells. These can be induced to fuse with protoplasm using PEG and have therefore been used for gene transfer. In this method, DNA enters the protoplasm due to endocytosis of the liposome. So, what we are doing when we are applying these liposomes, the process of endocytosis takes place and hence it involves the entry of the DNA into the plant cell. So, these are the steps. We have few steps which we have to follow. That the first step is adhesion of the liposome to the protoplasm surface. How can this be adhesive? By the fusion of the liposome at the site of adhesion. When we are introducing protoplasm, the particular site is there on which the liposome will attach itself. That particular point where the protoplasm, where the liposome is attaching, that point is called as adhesion point. So, the liposome will fuse on the adhesion point of the protoplasm. Then release of the plasmid inside the cell. Once the liposome is getting attached on the protoplasm, the liposome will release the plasmid inside the plant cell. So, liposomes possess many properties like those of biological membranes. They are easy to manipulate. The lipid content can be varied. Many substances may be trapped inside the interlamellar spaces. Example, liposome mediated transformation has been demonstrated in protoplasm of tobacco and carrots. Liposome mediated transformation showed higher efficiency when it was used in conjunction with the PEG, that is polyethylene glycol. Then stability and storage of nucleic acids one encapsulated. So, the advantages of the liposome mediated gene transfer is the protection of nucleic acids from nucleus digestion prior to introduction into the cellular environment, then reduced level of cell toxicity, then applicability to a wide range of cell types, then high degree of reproducibility. Because this liposome will make it sure that each and every protoplast is getting inserted with the foreign DNA, hence the high degree of reproducibility. All the protoplasts are able to be cultured in the lab, and each and every protoplast cell will divide and redivide to form multiple seedlings or multiple plantlings. So, hence delivery of encapsulated DNA by the membrane fusion, protection of nucleic acid from nucleus activity, delivery of intact small organelles. Even we are using this liposome process for the delivery of the intact small organelles like mitochondria or protoplasm or any other chloroplast cells inside the plant cells. And also we are using this method to protect the genetic material from immunogenic reaction. Liposomes are non-immunogenic. Then the third method is micro-injection. In this process, the genetic conversion, we are using the genetic conversion. How we are using genetic conversion? By injecting the DNA solution directly into the protoplast with the help of the micropipette. And this method was first discovered in the year 1987 by a scientist called Lichtenstein. At the time of injection, protoplast should be immobile. So, how we have to make the protoplast immobile? You have to fix the protoplast on a slide, and the slide has to be fixed under the microscope, so that you can see from the lens and you can inject the DNA solution directly into the protoplast cell. The injected protoplast should be capable of enough culture and new plant origin. The protoplast should be isolated from the meristematic tissue of the plant, so that it has the capacity to regenerate. Such type of protoplast can only be used. Otherwise, if we are using any other protoplast from any other portion of the plant cell, it may not be so producible. That is why we are using the protoplast cells, especially from the meristematic region of the plants. In this method, normally embryo cells or meristematic tissue or pollen grains are used, and with these parts of the plants can form the chimeric plants. What are chimeric plants? Chimeric plants are nothing but, in chimeric plants, the genetic or the DNA composition of the organ tissue is different in the different organs. If you go for mitochondria, or if you go for a chloroplast, or if you go for cells from other parts of the plant, then each and every organ of the plant will have a different DNA composition. So, we can make sure that with the help of the micro injection, we can maintain the same DNA composition. That is why we are using the micro injection as a method of gene transfer. Further, single cell culture technique used to produce plant. By this method, transgenic chimera of Brassica napus is produced successfully. So, this is one of the best examples where we can use this micro injection, that is the Brassica napus. So, we have some advantages and disadvantages. So, we will discuss about the advantages. This method frequency, its accuracy is more. The high approximate, that is 14% more than the electroporation or any other gene transfer method. For example, we have agrobacterium mediated gene transfer. In agrobacterium mediated gene transfer, we are using the TI plasmids of the agrobacterium, through which we are doing the gene transfer. What we are doing in agrobacterium, we are inserting the foreign DNA inside the agrobacterium, and we are infecting the plant with the agrobacterium. So, that this is also one of the direct methods. Here, because we are inserting foreign DNA directly into the agrobacterium, and we have to use this agrobacterium in our host plant. How we are using it? We have to make a small injury on the plant surface, on the bark of the plant. Then on that injury, you have to just put some bacterium, that is agrobacterium, along with the inserted foreign DNA, so that this agrobacterium will infect the plant, and from the TI plasmids of the agrobacterium, the foreign DNA will get incorporated into the DNA of our host plant. So, apart from that, in micro injection, the frequency is more, because we are using directly in the protoplast cell. Directly we are using the protoplast, we are fixing the protoplast of the cell on the slide, and we are fixing the slide in the under microscope, and from we are observing it from the lens, and we are directly inserting the DNA solution into the protoplast cell. So, the percentage of efficiency is more here when compared to the electroporation. In electroporation, the efficiency is less when compared to microinjection, because there in electroporation we have been using the high magnitude electric pulses, because of which most of the protoplasts are getting damaged. Here there is no question of the protoplast getting damaged. We are fixing the protoplast on the slide, and we are directly injecting the protoplast inside the injecting the DNA into the protoplast. So, the efficiency is more here. So, in this process the genetic conversion is done directly. So, this is the diagram. You can see that we are using the micropipette here. We are filling the DNA solution in the micropipette, and we are inserting it directly into the protoplast of the cell. So, in Brassica napus we can use this method of gene transfer. Now, the last process is particle bombardment. This method is also called as biolistic method, or this is also called as microprojectile method, or it is also called as shotgun method, or it is also called as particle gun method, because the principle here we are using is the principle of the gun. You know very well how the gun operates. We have to push the trigger, then only the bullet will come outside. Like that only the equipment what we are using for this method is exactly like a gun. The equipment is like a gun, and we are using the bullets here. The bullets are nothing but the particles, the gold or tungsten particles coated with the foreign DNA. These are used as bullets inside this equipment. This equipment is exactly having the principle of a gun. That is why we call it as a biolistic method, or we call it as a shotgun method or particle gun method. So, here we are using gold or tungsten particles coated with the foreign DNA, and these particles are also called as micro projectiles. So, that is why we also call it as micro projectile method. So, here what we are doing is we are accelerating these bullets. The bullets is nothing but the fine particles, the gold and tungsten particles which are coated with the DNA. So, these are called as bullets here. So, these bullets are being accelerated with high velocity, that is nearly 1400 feet per square per second. So, this is the velocity in which we are accelerating these bullets. So, when we push the trigger with the velocity of 1400 feet per second, this speed will push the bullets inside the barrel of the gun, and when we push it more, what happens is these micro projectiles are carrying the DNA with a lot of force, and they are getting bombarded with the protoplast cells directly. So, the size of the particles should be somewhere around 1 to 3 micrometers in diameter, and they are to be carried into the macro projectile. The name of the equipment is macro projectile, which is in the form of a gun. The principle is same as the gun. So, the various names of this particular method is particle acceleration method or bioblaster method, or it is also called as micro projectile bombardment, or it is also called as particle gun method, or this projectile method, or it is also called as the gene gun method, or it is also called as biolistics. In forensic medicine, the name, the technical term for the gun is biolistic. That is why we also call it as biolistic method. So, this method we can use in different experiments. For example, like gas entertainment, then transferred impulses and electrolytic acceleration, then macro projectile acceleration, then centripetal acceleration. Here we are using the centripetal forces of the particular equipment, and this is very most of the labs are not using this method because it is a very costly equipment. We have to use this gold or tungsten particles, then definitely it is cost effective. Most of the labs are not using it. You can see the structure here, the equipment which has got a trigger here at one end, and it is also filled with the DNA coated gold and tungsten particles, and from one side you have to fill the gas inside it, so that when you pull the trigger, the gas will put a lot of pressure on the particles or the bullets, and because of the pressure built inside the tunnel, the pressure, with a lot of pressure, it will move forward, and with a lot of thrust it can get inserted into the target cells. You can see at the bottom of the equipment, in a small petri dish, there is a protoplast present, and you have to just make it a target and push the trigger, so that with a lot of pressure these bullets will get inserted into the protoplast cells, and the DNA will get inserted in the protoplast, and the formation of rDNA takes place. And these are the several advantages and disadvantages. We have the several factors that might affect the efficiency of the foreign gene delivery. Morphological characters of the cells like the cell wall thickness or size of the aggregate cell, or the DNA carrying ability of the tungsten particle, or the effective damage. Sometimes what happens, because of the high force which has been inserted with the help of the trigger, what happens in a single cell, there can be insertion of more than one DNA particle, more than one particle. So, the expression, the degree of expression will be somewhat less than the other methods. Then advantages, you can see it is a rapid and simple procedure. It is a very simple procedure. It facilitates the direct transformation of the totipotent tissues such as pollen cells or embryo cells or meristems and morphogenic cell culture. It appears to be uniquely suitable for organelle transformation. Then thousands of particles are accelerated at the same time, causing multiple hits, resulting in transfer of genes in many cells simultaneously. So, here what is happening? Because we are pushing the cells or the particles with a lot of pressure, so thousands of particles are moving at the same time. So, simultaneously many particles are getting inserted into the target cells, and simultaneously you can see that each and every protoplast will have more than 5 or more than 10 bullets, that is along with the foreign DNA. Then disadvantages are it requires a special equipment which is not available uniformly or universally. In many of the labs, this equipment is not available because it is a cost effective, because it has to be provided with the gold and tungsten particles, which is again cost effective. That is why most of the labs are not providing this type of equipments. Then delivery efficiency is still not high. Delivery efficiency is not high because the lot of DNA particles are getting inserted into a single cell, because the bullets are not moving in a single direction. That is why it is moving in a particular direction where a particular few protoplast cells are present. So, lot of particles are getting inserted in few number of protoplasts. So, the efficiency of this particular mechanism is not so high when compared to electroporation or microinjection. Then potential users should be prepared to spend some time adapting then existing protocols to their specific species or tissue of interest. Thank you.